Okay, good morning, everyone. We are discussing about the chapter on translation. And in the last class, we learned some things about genetic code and the structure of tRNA and so on. So we'll continue that today. And I would want to start with something that some of the things that I haven't discussed in the last class. Firstly, I think we all have to remember the start codon and the stop codon. Right, start codon AUG and stop codons are already highlighted. Stop codons can also be called as nonsense codons, especially if in the context of mutations, you can call them as nonsense codons because they do not code for any amino acid. What I want to bring your attention here is to discuss a few things here. Especially, say for example, this is what I'm looking at this square. So you see CUU, CUC, CUA, CUG. The first and the second one are common in all these codons, CU, 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 CU. What is difference is the third codon. We will discuss about Obel hypothesis and so on at a later stage. But right now, what I want you to realize is that many tRNAs, one amino acid can be coded by multiple codons. Okay, so all these codons code for leucine. And there are also two more additional ones for leucine, UAA and UUG. So you can see that 6 different codons are coding for leucine. Okay, so similarly, you will also have many other amino acids that are being coded like this. Serine, here it is coded by 4. This whole box is for proline. And here it is for threonine, alanine, and so on. So keep in mind that multiple codons code for one amino acid, can code for one amino acid. So this is the degeneracy of codons. That means one amino acid could be represented by or encoded by multiple codons. So arginine is one of the other one which has 6 codons. Here is, and these are these 2, and so on. So why I'm bringing this up now is that if you're expecting arginine, for example, to have 6 different codons, then you're likely to also expect that there will be 6 tRNAs. So say, for example, arginine, and it is coded by 6 codons, then what do you expect? You would expect that there are 6 different tRNAs for arginine. If you expect 6 different tRNAs, it is also theoretically you would expect 6 arginine tRNA synthetases. That's not actually true, right? You may have these until here, it's not changeable. You have arginine, and it is coded by 6 codons, and you can have 6 tRNAs or even lesser. That we will discuss later on. But what I'm bringing here is about the tRNA synthetases. You don't need 6 different tRNA synthetases at all. You only need one tRNA synthetase that could recognize all these 6 tRNAs, right? And add arginine during charging. It will just add an arginine. We'll come to that. But let's quickly have a review of what we have learned. We have learned about the processing of the tRNA. You have 5' cleavage, 3' cleavage, 5' cleavage by RNase P, 3' by RNase D. This is specific a tRNA that is tRNA for tyrosine, the sequence and other details. And we also have CCA addition on the 3' end. That's an important, this is the location where the amino acid is going to be added. And we also have splicing. This part of the tRNA is removed out. And the mature mRNA, and also we have a lot of base modifications. We described about the methylation of several residues. We also discussed about dihydrouridine and also pseudouridine. We'll have a quick look. And then we will have the formation of a mature tRNA. And the modified bases, I wanted you to at least be able to remember what is pseudouridine and dihydrouridine. For that, you should have known the uridine, right? It's a pyrimidine ribose attached to the first carbon, 1' carbon. And in pseudouridine, instead of being attached to the, if you're counting the position of uridine, you will say this N, 1, position number 1. On the ribose, you will call it as 1'. So the ribose is, instead of being attached to the nitrogen in the first position, it is attached to the glycosidic bond forms in the 5th position carbon. Dihydrouridine is, it has 2 extra hydrogen bonds on 5th and 6th carbons. There is no more double bond there. There are many others, but we can give it a pass. Just try to know the names or things, at least the types of modifications. Then we also learned about the different arms of a tRNA. D loop, because it has a lot of dihydrouridine loops. You have pseudouridine loop, because it has pseudouridine conserved one. And there is the anticodon loop, which is, these are 3 bases here, which are the anticodons. Those are the ones that will base pair with the codon on the mRNA. So you call that anticodon. And the loop containing this anticodon is called as the anticodon loop. Then on the 3' side, we have this CCA. That is the site where amino acid is going to be attached. So we call that the acceptor arm. And variable loop has different sizes, lengths, and in some cases it's not present. And so we call it as variable loop. But otherwise, typically, majority of the tRNAs have these 4 arms, and their functions are almost similar in majority of the cases. And then we discussed about the charging of tRNA. That is how amino acid tRNAs are made. It happens in 2 steps. One is by adenylation, that is addition of AMP to amino acid. AMP is not so energetic compared to ATP. So the ATP, the O on the carboxyl group of the amino acid will make an attack on will bind with the alpha phosphate of ATP, and then it becomes an intermediate where from here to here it is the amino acid, and this part of it is the this part of it is the nucleic acid or nucleotide, right? AMP. So pyrophosphate is released, and the adenylated amino acid is now acted upon by the tRNA. Of course, all these things happen within the enzyme. We will discuss about it shortly. So the 3' OH, where we have the CCA, that attacks or binds with the carbon of the carboxylic group, forming, now you have a charged tRNA. That is amino acid bound to the tRNA with a high energy bond, and the AMP is released out. There are 2 classes of tRNAs. One class in which the amino acid is attached to 2' OH. In another class, it is attached to the 3' OH. Here we are talking about 3' OH of the tRNA. So this must have belonged to class 2 tRNA. Then we also discussed something about there are differences. This is what I have started with. 20 amino acids, 64 codons, and then we have how many synthetases are required. The minimum number required, or kind of in typically, is 20 tRNA synthetases. One synthetase can add one amino acid, one type of amino acid, right? tRNAs can be more. So if you had, we spoke of arginine. Arginine has six different codons, and probably the tRNA, that is the synthetase that is charging these tRNAs, can be one. Okay, it's not that it has to be one, but typically it can be one, because all it needs to do is be able to recognize that the tRNA is, say for example, this is the enzyme, and that tRNA is specific for arginine, and then you should add arginine to it, right? So the tRNA can be different tRNAs. So that's what is meant by they're iso-accepting. All the tRNAs that code for the same amino acid, or the adapters for the same amino acid, are charged by the same synthetase. We also had the how to read this. This is the amino acid that has been added. This is the superscript here indicates the tRNA that it is intended for. Okay, as you say, as you think of it, if you had always, you would want to, or it makes sense if the prefix and the superscript here, they should be same. Here there is a difference, and that is that is the example that we have discussed in the last class. In some bacteria there is no tRNA synthetase for glutamine. Then how does tRNA for glutamine get charged? The same enzyme recognizes both the tRNAs, that is for glutamic acid and glutamine, and it charges it with glutamic acid. Later, the second enzyme converts for those glutamic acids that are attached to the tRNA, the glutamine, they get converted into the glutamine. Okay, the difference is this amino group. An amino group is added to the carboxylic group of the extra carboxylic group that makes glutamic acid acidic. So it forms glutamine. So if you want to see it as a structure, you see this, all the gray one that is indicated in ribbons and sheets, that's the tRNA synthetase. The purple one represented here is the tRNA. And somewhere around here, you might be having the amino acid that is going to be added to the 3' end of the tRNA. So they are highly specific. The tRNA synthetases, they can bind specifically to tRNA by using the interactions between the bases and the amino acid side chains within the tRNA synthetases. So there is something called as discriminator base that is here. So here the sequence in this case is CCA, and there is a base upstream of the CCA sequence, and that's called the discriminator base. That can, say, if you change some of the tRNA synthetases use that base also for recognizing what kind of tRNA is interacting with them. So they can recognize based on, some of them interact with the anticodon loop, and also so that they can recognize what is here. But again, it will get complex for us because there are too many different tRNA synthetases to read, and they have different mechanisms as well. Right now, what you should remember is tRNA synthetases have mechanisms to figure out which tRNA is binding to it. The pocket for the amino acid is clear. That we will discuss in this slide. Say the difference between Tyrosine and phenylalanine is just this OH group, hydroxyl group, extra hydroxyl group is present in tyrosine. So you can think of how can, if you have a binding pocket like this for amino acid in a tRNA synthetase, the side chain here of phenylalanine is kind of hydrophobic. There are no charged groups there, and if that side group is present here, is inserted here, then it would make proper interactions. But if you had an extra OH, then it might have a different kind of interactions, right? Based on that, the active site can determine, based on the interactions between the active site of the tRNA synthetase and the side groups of the amino acids, whether the proper interaction is happening or not. And if there is, if you're talking about tyrosine tRNA synthetase, then it will have it will be able to make favorable hydrogen bonds because of the hydroxyl group on the tyrosine. So if valine entered into such, say for example, sorry, this is the pocket for tyrosine tRNA synthetase, TYR, and it has some base here that can, some amino acid group, side group that can interact with a hydroxyl group. Say I'm just putting it like this, tyrosine, okay, and this is the OH. Now the tRNA synthetase can, in a way, makes favorable interactions with this OH group here. Say for example, if phenylalanine entered, which does not have this hydroxyl group, then it cannot make those interactions. That means this is not tyrosine. Instead, it might be phenylalanine, and that enzyme is excluded out. Okay, so these are discriminating strategies or interactions that will allow the tRNA synthetase to figure out which one is the correct amino acid and which one is not. Another example is between valine and isoleucine. The difference between these two is just CH3. Why we are discussing, or this is an example, is in the previous case we discussed because it is a hydroxyl group, a polar group is added, and interactions are different. Here, valine and isoleucine both are hydrophobic. They do not, the side groups are hydrophobic. There is nothing to interact with. All of them are almost CH3. So in that case, how does it happen? I will try to draw for valine. Assume this is valine. I'm only talking about the active site. Okay, the rest of the enzyme is there. That's the valine pocket for valine tRNA synthetase. And I would want to also put for isoleucine, which is little bigger, right? So somebody, okay, will not get there. So valine can fit in here because this size, everything is matching. Whereas isoleucine cannot fit in here because the size is little smaller. For this, the methyl group, extra methyl group cannot fit into this one. So there is steric hindrance or steric exclusion. That is structurally, isoleucine cannot fit into the pocket made for valine because isoleucine is bigger. Whereas in this case, if you look at the isoleucine one, you can have valine also can go in, right? Well, I will tell you also, somebody was making a cat. I don't know whose story is this. I think it's Edison's or somebody, I don't know. He wants to have a cat house. He has two cats, one big one and one small cat. And so the scientists, sometimes they get crazy, I don't know. So he went to a carpenter or to a shop and argued that I want a big house for the big cat and a small house for a small cat. The shopkeeper or the carpenter probably was saying, why don't you just make the big house where both can fit in? And then there was a serious argument between those two for a while. I can't recollect the whole story, but that is how logic, right? It's the same logic in this case. You have a larger space, isoleucine can fit in there, and if isoleucine is not there, valine can also fit in there. But in that case, how does tRNA synthetase only use valine, sorry, isoleucine, and exclude valine? So it has to do with the binding of the, say, the binding energies of those two, which one is making a better interaction with those ones. And there is also one additional strategy that is called a binding editing pocket. Let me erase this once. Okay. Yes, I'll try to do it a little bit differently here, for one thing. So I will take a tRNA synthetase. Okay, I'll just putting it in a different orientation. So this is for isoleucine tRNA synthetase. And assume that tRNA is bound. Okay, we'll count this as tRNA. This is the 3 prime. And valine is bound here. Valine can also fit in the same active site with a lower energy probably. But still, it can fit in. And assume that the tRNA synthetase has added this one to the tRNA. The tRNA is actually specific for isoleucine, right? But it is added with valine. How do you write that? If you have to write, you will write val tRNA ISL or isoleucine. I'll just put it as ISL. Okay, that is wrong. This should not be there. So there is an additional pocket which is called as editing pocket somewhere around here. If the product the product or sorry, what has entered in, I forgot to mention that. Okay, anytime the ribosome, sorry, amino acids are entering into the tRNA, into the synthetases, they come as amino acid with AMP, right? Adenylated AMP, and then they are actually added to the tRNA. So if a valine, there is an editing pocket. Forget about this, what I said. Valine AMP is, there is a pocket for it, which can fit only valine. And if it enters there, it will break this bond between valine and AMP. So valine and AMP are released out because valine as such cannot be used for charging of the tRNAs. Okay, I hope you understood that. Yes, so it is twice proofreading. Once it is by the free energy based, because I'm talking only about isoleucine tRNA synthetase, because the free energy is higher for the interaction between the tRNA synthetase and isoleucine and lesser for valine. And secondly, there is an editing pocket within the isoleucine tRNA synthetase. That pocket, it only valine with AMP, charged AMP, valine AMP can fit into it, and if it goes there, the high energy bond is broken. So valine can no longer be used for charging a tRNA. Okay, that's how some of the examples for proofreading. This is the one that I'm now going to describe is not going to be immediately important for you in the exams, but I want you to know ribosome cannot see whether the correct tRNA is charged with the correct amino acid. The ribosome has only one duty. We will see it shortly in the next class. The ribosome is only responsible for the interactions between the codon and the anticodon of the tRNA. The ribosome cannot. So if this is 5 prime to 3 prime, this should be 5 prime and this should be 3 prime, and this is where the amino acid is attached. Ribosome is responsible for codon anticodon interactions and peptidyl transferase. The ribosome is not responsible or cannot discriminate if the correct amino acid is attached to the correct tRNA or not. Okay, that is what is the essence of this particular statement or slides. Okay, so say for example you take cysteine tRNA and you can do it by chemical reduction. You can remove the thiol group or SH here, and that results in alanine. Now here you see cysteine tRNA for cysteine. That's absolutely correct. But here, because of the chemical reduction, you have the cysteine is converted to alanine. Alanine tRNA for cysteine. So this tRNA is going to recognize codons for cysteine. And in that location where cysteine should have been added, alanine would be added. Okay, this is a artificial scenario that we are discussing. It is just to prove that the ribosome does not have any mechanisms to proofread whether the charging of the tRNA is correct or not. That's the job of the tRNA synthetases. After tRNA synthetases have done it and you made some kind of modifications, it's going to get incorporated in the places of cysteine, for example, in this case. Okay. So the next component is the ribosome. We all have pretty good idea about it in a way. First thing is, where does the translation occur? It happens in the cytoplasm, even in bacteria or in eukaryotes or in prokaryotes. Because in prokaryotes you do not have the nuclear membrane, so transcription, replication, everything also happens within the cytoplasm. And in prokaryotes you have transcription and translation coupling, in the sense that transcription is happening this way by RNA polymerase, mRNA is being synthesized, and translation already resumes on the mRNA. So that is what you call it as coupling of transcription and translation. So there is a certain speed. It adds 20 amino acids per second. That's a huge thing. It's not the same as it is very less compared to that of transcription or replication. There it is different. You have a template, you have to put a block here and keep on polymerizing. But ribosome is different. You have to read something else and then make something else out of it, right? So ribosome, 20 amino acids per second, I think is still great and huge. Yes, and then in eukaryote, the rate of translation is little slower compared to that of prokaryotes. In majority of the prokaryotes, if not all, speed is everything. You can make errors, but be as fast as possible, because the competition is heavy and the resources are very limited. In eukaryotes, the scenario is little different. We can eat and starve for a day, and then, or starve for 3-4 days, and then eat the next day and still survive. Bacteria, that's not a scenario. If you don't eat for an hour or something, you're starting to die. Whereas in higher organisms, complex multicellular organisms, even if you don't eat for, I think a human can starve up to 15-20 days, I think. All we need only two things that we cannot stand without, we cannot live without, is air and water. Without air, minutes. Without water, maybe a day or 2 days. So we need those things supply. Okay, so in transcription, why it has to be coupled, transcription and translation should be coupled, and why is the rate so high? These are some of the explanations. So the ribosomes are measured in they are 70s in prokaryotes, 80s in eukaryotes. They have two subunits. One is called the large subunit, and the other one is called as the small subunit. So the large subunit in prokaryotes is 50s, and the small subunit is 30s. In eukaryotes, I think it is something like this, 60s and 40s. So what is this S? That's called Swedberg units. It's a rate of or coefficient of a rate of sedimentation. So the larger the S value, the larger the molecule. Okay. So what is the biggest function of this large subunit and biggest function of small subunit? In the previous slide, I described that ribosome has two functions: to read the mRNA, and second one is to make the protein. That is, the smaller subunit is mostly involved in reading the mRNA, decoding center, you call it. And the large subunit is the one that makes the protein, and that happens because of the peptidyl transferase center, the reaction of making amino acids, polymerization of amino acids. So each subunit again has, say, for example, in this case, eukaryotic ribosomes. They are 80s. The molecular weight is very high, and they have 60s subunit and a 40s subunit. Each of them have several proteins. Say the large subunit has up to 49 proteins and several rRNA molecules, right? So 28S rRNA, 5S rRNA, and 5.8 srRNA. The small subunit has about 33 proteins and then 18S rRNA. Prokaryote also are very similar, but they're a little more simpler. The small subunit has 21 proteins and something 16S rRNA. So you can see that 1500, approximately 1500 nucleotides. And the large subunit has 23S rRNA and 5S rRNA and about 34 proteins. Okay, so you can remember that if the to me the evolution of ribosomes is something, I might have said, is something puzzling. So you need to make a ribosome, and ribosome has a lot of these proteins. It has the RNA molecules. So that means the ribosome that we know of in the current world right now, it's billions of years. They need the RNA component, and they also need the protein component. And as we go back in history, in long ways, billions of years, the ribosome probably is made just of the RNA molecules, because the first proteins to be synthesized would have required ribosomes, isn't it? Ribosome is the one that makes the proteins. Okay, and in that case, how can a ribosome come into existence with all the proteins already? That's not going to happen. Instead, there could have been 3 or 4 long RNA molecules folded upon themselves, which which might have given rise to, eventually given rise to, the formation of the ribosomes, structurally the way the ribosomes are there in the current day. So the earliest ribosomes must have been made up of completely rRNA, and they might have had the pockets of reaction centers to read the mRNA, to make the peptidyl transferase reaction. Even now, if you see it this way, um, the interior of the ribosomal subunit is covered with a lot of protein, a lot of RNA, and you will see that the outside you will see many of those proteins. I mean, what I'm trying to say is the centers of decoding, and the centers of peptidyl transferase. The functions that we described for small subunit is decoding, and the important function for the large subunit we said was peptidyl transferase center, PTC. If you see the RNA component, these two important reactions, they both are, the majority of the work or the participants are from the rRNA. And that is how some people also think ribosome as a ribozyme. Why is that? Because the important things, events that are happening within the ribosomes are still carried out by the rRNA components. Okay. Anyway, it's a good way of trying to visualize how things have happened over time. What is a polyribosome or a polysome? So, you, if there is an mRNA, you have 5 prime to 3 prime here, and assume this is an eukaryotic mRNA, or so you'll have a start codon and you will have multiple codons continuously, and then you will end up with a stop codon. That's an open reading frame. Sorry. Then ribosomes, we also discussed about something called as ribosome binding site. In prokaryotes, it's called as SD sequence. In eukaryotes, it is called as Kozak sequence. So the ribosomes can only bind here to start with. They bind and then they start translating the mRNA. So the ribosome will keep moving about like this. And I think you have to also remember that the contacts that the ribosome covers is about say 30 bases, and but the subunits are actually very large. The ribosome is large, so it occupies a space, or you can expect one ribosome every 80 nucleotides, right? Something like this. From here to here, it could be 80 nucleotides. This could be another 80 nucleotides. The actual contacts that the ribosome is making is only 30 nucleotides. So is a ORF of 1,000 base pairs, 1,000 bases could accommodate about 10 ribosomes. So what is a polyribosome? One mRNA loaded with multiple ribosomes is called a polysome or a polyribosome. And we'll also look at the chemistry of peptidyl transferase before we dive into what the mechanisms of translation. The peptidyl transferase reaction, or this peptidyl transferase reaction happens in peptidyl transferase center, that's in the large subunit. So what happens is that we will discuss again when we come to elongation also, and also in initiation, but you have to remember that peptidyl tRNA and amino acyl tRNA. I would want to describe that here. Yeah, it will be here, but just remember the chemistry part only we will discuss. So this is the one of the tRNAs here, and this is in the other place of the ribosome. There are 3 sites. This is called the P site, this will be called as the A site, and this one is the E site within the ribosome. Okay, so this is the incoming newly entered tRNA. This is where the peptide chain is growing. Here you had one amino acid, CONH, that's the peptide bond, and this is the next amino acid. So the carboxylic group that is present here will break and will join with the NH2 here. That is where new peptide bond has formed. And this chain, this tRNA becomes free, and upon translocation, this goes here, and that will be eliminated eventually. Okay, so what you need to remember is translation happens from N to C. N is for amino group, C is for carboxylic group. Or you can also say in words amino group and carboxylic group. Why is that? This carboxyl group is attached to this one, and this is going to be added to this. That means on top of this, and in this end, you will see that it is N. As an overview of translation, if you are going to look at the previous picture, that should be much more helpful, because you see the reading of by the ribosome of the mRNA happens 5 prime to 3 prime. Majority of the reading everywhere of nucleic acids happens from 5 prime to 3 prime. And this is what does this say? There are three. This is a protein that is growing polypeptide. That means it has three amino acids here. That means it has read somewhere three codons so far, right? As the ribosomes are moving this way, they will read more number of codons, and more the polypeptide chain will keep growing, right? And the first base that is going to be added sorry, first amino acid that's going to be added, I'll try to put tRNA like this, and this is the 3 prime end of the tRNA. You have N-formyl methionine that is attached. That's the initiator tRNA. And how is it attached um, to the tRNA? Through its carboxylic group. What is free? It is the amino group, and this will move to the next one during peptidyl transferase. We'll see it in the later class. So as a overview of translation that we are going to start, we will have it in three pieces, that is initiation, elongation, and termination. So we will initiation happens here. Along with the small ribosomal subunit comes and binds to the mRNA, and along with the then we have the initiator tRNA binding at the P site, and then the large subunit attaches to the ribosome and makes a full ribosome. Then more amino acyl or charged tRNAs will keep coming in as the ribosome will keep moving, and that is called as translocation when it moves, and it's also in this case elongation. More amino acyl tRNAs will enter, and peptidyl transferase reaction happens here, and this continues until the ribosome reaches a stop codon, and that is where termination occurs, where the ribosomal subunits are released, the newly synthesized protein is released, and the mRNA is released, right? So what we will, from here to here, we will discuss it as initiation, or maybe we should go till here. This is initiation, and from to here as elongation, and then termination. I will take initiation, and then I will stop. So as I was discussing, the ribosome can be arbitrarily made into three parts, which is called as three sections, that is A, P, and E sites. A for amino acyl tRNA, that is the entry of tRNA happens in this case. This is the peptidyl tRNA site, where the growing polypeptide is usually happening here. And then you have the exit site, where the tRNA, once it comes here and leaves the ribosome. Sorry. For the initiation to happen, we first require the recognition of the ribosome, a recognition of the mRNA by the ribosome, and that happens by the rRNA component of the small subunit. In this case, 16S rRNA, we are talking about prokaryotes. In eukaryotes, that is 18S rRNA. So the recognition of mRNA, you have this site, the ribosome binding site, or you call it as the SD sequence in prokaryotes, Shine-Dalgarno sequence. In eukaryotes, it is Kozak sequence. So you have the ribosomal subunits, there is mRNA, and there is initiator tRNA. And so the first is the small subunit binds to the mRNA by recognizing the SD sequence and the start codon. So there is SD sequence, there is a spacer, and then you have a start codon. So based on that, these three components come together, on top of which you have the large subunit attached to it, right? So that is how the initiation actually happens in prokaryotes. It's simple, but it requires many factors. But to start with, we'll have to discuss about initiator tRNA. What is the codon? AUG is the start codon um, for any of the ORF open reading frames, and it codes for methionine. So AUG can be within will be at the start, and it can be, say, assume this one is a codon, this is another codon, this is another, and you can have a codon inside also. This is another codon. The tRNA that recognizes this will code for methionine. Okay, we'll add methionine there. The tRNA that recognizes the AUG as the start codon. This is not the start codon here, right? This AUG is not a start codon. This is the start codon, because it's beyond that you do not have. Instead, you actually have the ribosome binding site here. Okay. So that is called as tRNA I for initiator tRNA, and it is charged with N-formyl methionine. What is N-formyl methionine? On the amino group, you have an aldehyde like this, or CHO. Sorry, that's formyl group on the amino group of methionine. That is called as F-meth. And the initiator, this recognizes AUG, and the second one also recognizes AUG, but this is the initiator tRNA. It's a different one, and this is normal one. Okay, so the initiator tRNA is always will always have this N-formyl methionine. And once the protein is made, now if, say, for example, a protein is made, and the first amino acid that usually it has would be the F-meth, right? F is not a good thing. The formal group is not required. So there might be, upon completion of translation, there might be enzymes like deformylase, which might remove the formal group, or you may have aminopeptidases. If you learned about protein targeting in cell biology in your in your previous semester cell biology, how proteins are targeted to a lysosome or somewhere to endoplasmic reticulum, or to be secreted out to the extracellular side. Each of those proteins have some kind of addresses on their N terminus. I hope you remember something called as nuclear localizing signal, which means those proteins should be sent to the nucleus. So these are the signal peptides that are present in the N terminus of the proteins. Once the job is done, usually these peptides are cleaved away by aminopeptidases. So the N terminus, in many cases, if not all, the tips of the proteins are the end on the N terminus. They are usually removed. Some 2 or 3 amino acids. Sometimes they can be even larger fragments of protein as well. So I hope you understood the initiator tRNA. It is a special tRNA that is the one that starts required for initiation to happen. And it is for N-formyl methionine. It has a formyl group on the amino group of methionine. Okay, I will take the things in the next class. Yeah, we'll start initiation, elongation, and we'll complete termination as well in the next class. So I suggest you to have a look at the textbook, at least look at the figures and read the legends, so that you will feel you will be at a greater advantage. Okay, thank you very much. I'll stop the recording.